Okay, we have chapter 3, the cell.
Okay, this is the cell. First, we have the plasma membrane. It separates two of the body's major fluid compartments, the intracellular fluid within the cells and the extracellular fluid outside. Then we have the cytosol, which is the fluid cytoplasmic material where many organelles are suspended. These are free ribosomes, and these are membrane-bound ribosomes called rough endoplasmic reticulum. It is the site of protein synthesis. Then we have the mitochondria. It is the site of ATP synthesis. Then we have smooth endoplasmic reticulum. It is the site of lipid synthesis. Then we have Golgi apparatus. This packages, modifies, and segregates proteins for secretion from the cell.
Now we have mitosis.
This is mitosis. First, we have prophase. The nuclear membrane degenerates and the chromatin condenses into individual chromosomes, and the mitotic spindle migrates on the opposite poles.
Then we have metaphase. It's when the chromosomes line across the center of the cell.
Then we have anaphase. The chromatids separate and they are pulled onto opposite sides of the cells.
Then we have telophase. It's when the nucleoli reappear within the nuclei, and the spindle breaks down and disappears, and this is the cleavage fertile.
Now we're going to talk about membrane junctions.
We have membrane junctions, and then we have tight. Tight junctions are a series of integral protein molecules in the plasma membrane of an adjacent cell fused together like the zipper of a Ziploc bag. Tight junctions help prevent molecules from passing through the extracellular space between adjacent cells and restrict the movements of membrane proteins. Tight junctions also form the impermeable junction that encircles the cell and separates one fluid-filled compartment from another. Then we have anchoring junctions. Anchoring junctions are also called desmosomes. They are mechanical couplings scattered like the rivulets along the sides of adjacent cell to prevent the separation. Anchoring junctions are also really strong connections between cells. Then we have gap junctions. Gap junctions is a communicating junction between adjacent cells. Gap junctions are composed of different transmembrane proteins, and they determine what can pass through them from one cell to another. Gap junctions. That's it.
Now we have tonicity.
Okay. We have tonicity. Tonicity is the ability of a solution to change the shape or plasma membrane tension of cells by altering the cell's internal water volume. First, we have isotonic solution. Isotonic solution is when the cells retain their normal size and shape in the isotonic solution, which is the same solute and water concentration as inside the cell. It's when the water moves in and out of the cell. Second, we have hypertonic solution. It's when the cells lose water by osmosis and shrink in hypertonic solution. It's when it contains a higher concentration of non-penetrating solutes than seen in the cell. For example, a strong saline solution. Last, we have hypotonic solution. Hypertonic solution is when the cells take on water by osmosis until they become bloated and burst. And it contains a lower concentration of non-penetrating solutes that are present inside the cell. Cells placed in a hypertonic solution plump up rapidly as water rushes into them.
Now we have movement of materials.
Moving on, we talked about that endocytosis moves things into the cell. And then first we have phagocytosis. Phagocytosis is cell eating. It's when the cell engulfs some relatively large or solid material, such as clumps of bacteria, cell debris, or inanimate particles. For example, fiber or glass.
Then we have pinocytosis. In pinocytosis, it is called cell drinking, also called fluid phase endocytosis. A bit of enfolding plasma membrane surrounds a very small volume of extracellular fluid containing dissolved molecules Unlike phagocytosis, pinocytosis is a routine activity of most cells, affording them a non-selective way of sampling the extracellular fluid.
Last, we have the receptor-mediated endocytosis. It's when the main mechanism for the specific endocytosis and transcytosis of most macromolecules by body cells. This exquisitely selective mechanism allows cells to concentrate material that is present only in small amounts in the extracellular fluid. The receptors for this process are plasma membrane proteins that bind only certain substances.
Next, we have movement of materials continued.
Transport requires transport proteins that combine specifically and reversibly with the transported substances. Active transporters move solutes, most importantly ions, uphill against a concentration gradient. To do this work, cells must expend energy. Next, we have vesicular transport. It's when the fluids containing large particles and micromolecules are transported across the cellular membranes inside bubble-like membranous sacs called vesicles. Like active transport, vesicular transport moves substances into the cell, which is endocytosis, and out of the cell, which is exo. Endocytosis is when the vesicle transports that eject substances from the cell interior to the extracellular fluid. Exocytosis is typically stimulated by a cell surface signal such as binding of a hormone to a membrane receptor or change in membrane voltage. Endocytosis is when vesicles provide the main route for bringing bulk solids, most micromolecules, and fluids into a cell. Many types of endocytosis rely on receptors in the membrane to determine the substance to be transported. So basically, exocytosis is when you move things out of the cell, and endo is when you move things into the cell.
Next, we have protein synthesis or gene action.
Now we have protein synthesis or gene action. RNA is single-stranded. It is ribonucleic acid. Now we have messenger RNA, which is mRNA. It is relatively long nucleotide strands resembling half DNA molecules, one of the two strands of a DNA molecule coding for protein structure. mRNA carries a transcript of the code to the cytoplasm, where the protein synthesis occurs.
Next, we have transfer RNA, which is tRNA. It is small, roughly L-shaped molecules that ferry amino acids to the ribosomes. They are decode mRNA's message for amino acids sequence and the polypeptide to be built.
Lastly, we have ribosomal RNA, which is rRNA. Along with proteins, forms the ribosomes, which consists of two subunits, one large and one small. The two subunit types combine to form functional ribosome.
Now we have membrane lipids.
Okay, now we have membrane lipids. This is the phospholipid bilayer. It has two layers, the polar head, which is phosphate head. It allows the head to interact with H2O. Then we have nonpolar hydrophobic tail, and that's lipid tails, and they hate water. The phospholipid bilayer makes up plasma membrane and organelle membranes.
Then we have the polar hydrophilic head. It faces the water inside and outside the cell and includes the phosphate group. Polar head hydrophilic heads of phospholipids are attached to water, the main constituent of both the intracellular and extracellular fluids. As a result, they lie on both the inner and outer surfaces of the membrane.
Then we have nonpolar hydrophobic tail, and they hide from the water. They are fatty acids. The nonpolar tails of phospholipid begin being hydrophobic, avoid water, and line up in the center of the membrane.
Next, we have cholesterol. Like phospholipids, cholesterol has a polar and a nonpolar region. It wedges its plate like a hydrocarbon ring between the phospholipid tails, which stiffen up the membrane.